Let's see, so clonal reproduction in Panama, Kylar's group found that it could generate up to 74 kilograms per month per hectare. So we expected to kind of see some of these clonal genotypes, which in this chart you'll be able to see. Some genotypes were even exceeding 50% of the population sample that we took. You can see here, I have it listed as a Wahoo here, the number of different genotypes that were clonal and how much of the total sample that made up. And there was often one really common clonal genotype and a number of other ones that occurred maybe twice or three times. You can see here, Molokai was one of the sexual reproducing populations there next to Ali'i Fish Pond, and had much less clones and really had a lot of genetic diversity in that population. It was interesting. And you can see there the Liliopolani Gardens is pretty much just one individual. Also another population, a mile or two down the road in Hilo, was only composed of a handful of genotypes, and there were only 2 alleles present at every locus. For our loci, we had in general, low polymorphism, so one marker only had 2 different alleles, but we had 2 that had 5 alleles. So let's see, I had to, I guess, separate some of these duplicate genotypes to get some accurate kind of statistics there, and also remove the haploid genotypes from the list. So I had some pairwise FST. There you can see that populations have a wide variety of similarity or distance. Populations like Molokai definitely form a group. Oahu, which was the island of introduction, has the highest allelic diversity among all of the islands, and there's an allele that's specific actually for this island, not observed in any of the other ones. Maui, one population from West Maui is Kahana, and the other is Kanaha, which is over towards Kahului, were not genetically similar, and Kahana appears pretty isolated, had some unique alleles, and was differentiated from most of the other populations by a lot. Molokai, the 2 populations there share alleles, and also seems to share a handful of alleles at 2 loci with Oahu. And then the Big Island has the fewest alleles, only 2 per locus, a lot of clones, and doesn't really have many populations established on that island. Kauai actually just got the results back and are currently processing those. So, let's see. There's a great variety of the gamete or spore-producing thalli at each collection. Interestingly, as I said, a lot of plants produce tetraspores in great amounts. They can continue to produce them over and over, yet those would germinate and produce haploid gametophytes, yet they're very rare. And even if they weren't fertile, we don't see very many genotypes that are composed of all homozygous alleles, which you would suspect would be gametophytes. When we do collect gametophytes, there's several in a population, so we think that it has something to do with environmental conditions rather than the season. So the results and the groups of genetic variation that we saw with microsatellites kind of substantiated a pilot study we conducted with markers similar to rapids 2 years ago. So we'd like to be able to answer a lot of these questions, and I think as we continue and add more data and make some specific collections, we'll be able to. But it was definitely multiple genotypes we think that were introduced or ongoing. Soon we're going to add in the samples from microsatellite genotyping from around the Pacific. Inter-island dispersal, pretty sure that spores and gametes are not able to make it through the canal, the different channels, sorry. Believe that hull fouling would be responsible for any of the inter-island transport. Possibly those rafting fragments that we'd see. It would be interesting to collect some future collections directly from inside of boat harbors or on hull-fouled boats to really see exactly what's going on. As I mentioned earlier, we think there's some kind of transport between Molokai and Oahu, since they share some rare alleles. So our population structure is interesting. The sequence markers probably didn't have enough resolution, but the microsatellites revealed a lot. We hope people will inform resource management. It's very inexpensive now, $1.50 to genotype an individual. So we'd be able to identify the origin maybe of new populations if we really build up this database, and hopefully be able to provide guidance in selecting populations to be targeted if there were going to be removal efforts. Some populations appear to be really new, composed of only a couple of genotypes, and hopefully that would reflect that they'd not been long established and hopefully not damaged, other aspects of the ecosystem. And so if, removed, maybe a slightly more positive response. And hopefully we'll be able to make some generalizations from this data that, a lot of red algae are the ones that are invaders, and these patterns of dispersal might be used to be able to predict the path maybe, or at least better than knowing nothing, like as we do now, about dispersal throughout the island. So, future, we definitely going to continue on with this project and hope to develop a whole new set of microsatellite loci. As I said, we have 7 now. We'd like to get at least 15, and we have libraries to work from. We want to make new collections to test some specific hypotheses regarding dispersal and barriers to gene flow, add in the samples, as I said, from the Pacific and one from the Caribbean, and really get some advice or some sophisticated analysis that'll be appropriate for the mating system that's found in the species. So I guess just the acknowledgements, HCRI, DAR for all their assistance, and various collaborators at UH.